Hi, my name is Lara Brindisi, and I'm a PhD candidate at Rutgers University in Dr. Jim Simon's lab, and I'm going to be demonstrating how to create a ddRADseq library. This method was originally developed at Cornell University by Poland et al. In 2012 and was later adapted for sweet basil by Pine et al. In 2017 at Rutgers University. Okay, so first off, what is ddRADseq? Well, it stands for Double Digest Restriction Site Associated DNA Sequencing. Okay, that's a mouthful, but there's a lot of really important information in the title. And essentially what it's referring to is that we are using two specific enzymes to reduce the size and the complexity of the genomic DNA. So the next question is, why do we use ddRADseq? And the answer is that we can't just extract a full plant genome and send it off for sequencing as is. We need to first process it into a library. And ddRADseq is really attractive to plant biologists because it allows us to reduce the size when plant genomes tend to be huge. It allows us to reduce the complexity because plant genomes tend to be really redundant. And it also makes it more affordable. We're able to pool our samples at the end, so that way we can sample 48 samples at a time instead of just one, reducing the cost 48-fold. Now, some labs may pool up to 96, they may pool less than 48. We found 48 optimal for our lab, and that's why you'll see me working in half plates. The protocol also enables us to do other important library processing techniques like quality control and cleanup and concentration adjustment. Now, the first step after DNA extraction is diluting the DNA of all the samples down to the same concentration. I typically aim for 50 nanograms per microliter, but you can go as low as 20 nanograms per microliter as long as they're all the same concentration. And the way that I keep track is I develop a template in advance that has all of my sample names, what plate they're going onto, what well they're going into, and exactly how much DNA and how much water to add based on the original concentration using the formula C1V1 equals C2V2. Now, the hardest part of GBS is staying organized and paying attention and staying focused. When I add the water and the DNA, I'm constantly talking to myself and saying exactly what I'm doing in my head as I do it. The first step that I'll do is add all of the water. I make sure that it's DNase and RNase free. And really now the biggest holdup is changing all of the numbers on my pipette. Let's get started. So now I like to check to make sure that all of my pipetting looks good. I take a look at the bottom of the well, make sure that the amount looks correct. And if so, then I'm ready to go on to the next stage of adding in my DNA. Now, the DNA is a little more risky to add than the water because you don't want to risk cross-contaminating your cells. So I like to work one row at a time while keeping all of the rest of my DNA on ice. I also cap it after every row. So now I check my concentrations, make sure they're all the same. It is a little hard to tell because there are some bubbles in here. So fortunately in this lab we have a plate spinner, so I'm gonna go spin those down. So now all of the levels are even. So we have done the dilution correctly. And now this plate can be stored in the freezer for future use. The next step is a restriction digest. And in the case of ddRADseq, DD standing for double digest, we use two enzymes. The ones that we found to work on sweet basil was MSP1 and PST1HF. So those will get added to our master mix. Now what goes into our master mix is adapted to our concentration of DNA. Since I concentrated my DNA down to 50 nanograms per microliter, I will be adding 22.2 microliters of nuclease-free water, 3.0 microliters of 10x NE buffer 4, 0.4 microliters of each enzyme, which we keep stored at the freezer, so that way we keep them at really cold temperatures the whole time. And then we add 200 nanograms of genomic DNA, which in my case is 4 microliters. I've calculated my master mix so that way I have enough for every single well in addition to a little bit extra so I can put it into the sterile basin and then use a multi-channel pipette. So I'll mix it up now and then add the amount, a total of 26 to every single well. And now I add my genomic DNA. I have to change tips every time because each sample is different. And I like to use filter tips for this just so that it doesn't risk contaminating my original stock DNA. Then we seal the plate with a microseal. Lastly, we spin the plate down to remove any air bubbles, and we put it on the thermocycler at 37 degrees Celsius for 2 hours. All right, so we are ready for our ligation step. We have a few important things here. 1, our restriction digest product from the previous step. 2, all the ingredients for our master mix. And then 3, we have barcode adapters. Now these have been previously diluted and placed in a 96 well plate. And each of these wells contains a unique oligonucleotide sequence that is meant to bind to one end of our digested fragments. This is a really important step because these adapters will each correspond with a different sample. So I could then use these barcodes later on in the sequencing processing to be able to determine that on plate 1 in cell A1, all of those DNA fragments belong to sample OCB02. So this is really important because at the end, we're going to be pooling all of our samples into one tube, and we need a way to be able to identify which samples those pieces of DNA belong to when we get our raw information back from the sequencing company. And then also on the other end of the fragment, so we have the barcodes on one end of the fragments that we've digested, and then on the other end we have our common adapter. And what the common adapter does is it relates to our primers that we'll use in the PCR step, and it will amplify based on that. Okay, so the first part is we'll make our master mix with all of the ingredients except for the ligase. The ligase is really sensitive to temperature, and it'll take a little bit of time to put the digest and the adapters into our 96 well plate. So we're going to add our master mix actually in the later stages so that we can have our ligase sitting in the freezer for as long as possible. But we can prep the rest of the master mix in the meantime to save some in-between time. So in each well, we'll have 11 microliters of nuclease-free water, 4 microliters of ATP, 1.5 microliters of our common adapter, and then 2 microliters of our NE buffer 4. And we multiply all of that by 54 because I have 48 samples here plus an extra 6 to allow us to work in the multi-channel pipette. So now we have our master mix. We're not pipetting it into every well yet, but we're going to put our digest into every well, 20 microliters. So then we add 1 microliter of our barcode into every well. It's very important not to mix this stage up, as I guess it is important with every stage. But we also want to use filter tips if we can, so that way we don't risk contaminating our original barcodes, because we use this for every single plate. Okay, now that we have our digest product and our barcodes in our plate, we can add our ligase to the master mix and pipette that into every well. So then we seal the plate with the microseal, spin it down to remove any air bubbles, and put it on the thermocycler at 22 degrees Celsius for 2 hours with a 20-minute heat stage at 65 degrees Celsius to deactivate the ligase. For the cleanup step, there's a couple of solutions that we need to prepare in advance, the most important of which being the AM Pure XP beads. Now these are magnetic beads that the DNA adheres to, and they kind of look like mud. They're sensitive to light, so we keep them in tinfoil. They also settle out really easily, so we actually will vortex them for 1 minute in the beginning, and then every 4 samples. Because they settle, we cannot use a multi-channel pipette, so we have to pipette them each individually. Now this step uses a lot of tips, so I try to minimize the amount of tips that I can use where possible, but you don't want to risk contamination, and you do need to mix very thoroughly, so you need to go through many boxes of tips, unfortunately. The other solution is fresh ethanol. So this has to be 200 proof molecular grade ethanol. We make 70% fresh every time that we do this protocol. Then after that, the first step is to vortex our beads and add them to every well. We add 20 microliters. So after we've mixed all of our beads into the solution, we let it incubate at room temperature for 5 minutes. Now, what I forgot to mention before about the beads is these beads have been adjusted to concentration that is appropriate for our method so that it can size select the DNA that we want. Now, this needs to be developed in every lab before you develop this protocol. And then these are aliquoted out into bottles that we know come to room temperature after waiting 45 minutes. So before starting this protocol, I brought them down to room temperature and then vortex for 1 minute. The next few steps are definitely the most tedious of the ddRADseq pipeline. What we're doing is we are removing the supernatant and going through a series of ethanol washes to clean the DNA fragments. And it is tedious because we need to remove all of the supernatant and then all of the ethanol completely. So first we'll go in with the larger tips, the 200 microliter tips on the larger multi-channel, remove the bulk of the liquid, and then we go in with the finer tips, the 10 microliter tips, and remove any excess of the supernatant. We go through 3 ethanol washes with the 70% ethanol that we made outside of this video in the well. And then we have to remove all of that similarly to the way that we removed the supernatant. And then we allow it to dry completely because ethanol can really interfere with the next step, which is PCR. Now I let the ethanol dry out completely. Sometimes this is a little bit difficult to do, so if we really need to, we can go in with these sterile cotton swabs and we can wipe out extra parts of the ethanol. We do this for approximately 5 to 10 minutes. Sometimes it takes 10 to 12 minutes. It really depends on the conditions in your lab. We put it in the fume hood as well to kind of speed up the process and add a little bit of extra aeration. Okay, taking a look at our plate after 10 minutes. It looks like there's still a few wet spots. So I'm gonna go in with the cotton swabs and remove them. Being careful not to touch them because they are sterile. So now they look pretty dry. I don't know if you can see in the video, but they look like they're cracking a little bit at the bottom. And there's no droplets along the edges of ethanol. So we can go ahead and now dissolve them in what we call EB buffer by QIAGEN. We add 40 microliters. And this can be a little bit tough. Again, this is another part of the tedious process. But what we're looking to do is pipette 40 microliters into each well of the EB buffer very gently, and then get these beads back into solution. There's a couple different ways. The recommended way is to pipette the solution into every well and then remove from the stand and then dissolve them in solution. This can be a little challenging and can take a little bit of time. They'll often be flaky, but you want them to be really homogeneous in the solution. And I'll show you an example. Hopefully you can see it in the video. I like to take the whole thing off the stand at this point, off the magnetic stand that is, because they're pretty stuck to the wall. And then I add, and this way it minimizes pipette tips. At first you can see it's flaky. Like, it's still mostly clear. Hopefully you can see that, I don't know. It's mostly flaky and clear. You want to mix it until it returns to that mud-like consistency that we saw in the beginning. So that is, in the first row, that is a more appropriate consistency. And that means now that they're in solution. Sometimes the tips fall off of the pipette because you kind of got to rub it around the well to be able to get them to go back into solution. You just push them back up. That takes a while sometimes. But then what we do is we put it back on the magnetic strip now that we've mixed the DNA back into solution. We want to still remove the beads and get just our clean DNA. Okay. After this sits on the plate for about 1 minute, we pipette as much of it as we can, in this case 37.5 microliters of our clean DNA into a new plate so that we can use that for PCR. And that's the end of the step. Our next step is to amplify the clean DNA from the previous step using PCR. And the way we do so first is by creating a master mix. And in this master mix, for every well, we'll have 16 microliters of nuclease-free water, 5 microliters of 5x TAC master mix, and 0.5 microliters of each primer, primer 1 and primer 2. Now these primers are designed for amplifying the common adapter in the ligation step. So again, they're also multiplied by 54 so that I have enough for all 48 of my samples, and then a little bit extra so that I can use the multi-channel pipette. Okay. And now we have 3 microliters of our clean DNA. So then we seal it with a microseal, spin it down, and then put it on the thermocycler. Now the settings for this PCR amplification are 95 degrees Celsius for 30 seconds, 16 cycles of 95 degrees Celsius for 30 seconds, 62 degrees Celsius for 20 seconds, 68 degrees Celsius for 15 seconds, and then 68 degrees Celsius for 5 minutes, and then we hold at 8 degrees Celsius, but promptly remove. Now we have to determine if our pipeline worked. And the way that we do that is we evaluate each of our PCR products' concentration on the qubit fluorometer. And what this does is it uses a double-stranded reagent for in each of the samples and compares it against that within the standards to calibrate the instrument. What we'll do is we'll make a working stock solution where we have a ratio of 199 microliters of buffer to 1 microliter of double-stranded DNA reagent. And I'm leaving this in the box until we use it because it is light sensitive. That's also why this is tin foiled. We mix that up and we pipette 196 microliters into every one of these little qubit tubes. We then add 4 microliters of our DNA into each of these tubes. We let it incubate for 2 minutes. But before we can actually get our readings, we have to calibrate it using a standard. Now, these standards are in the refrigerator, so I'll go and grab those and put them in immediately, wait 2 minutes, get a reading, and then I can start going through the pipeline, and hopefully it's successful. And I write all of these numbers on a plate record. So here I've already written down every one of my samples, along with their unique barcode adapter, so that if I have to redo any of these samples, I can immediately know what barcode I have to put in, what sample it is, and what well it's going into. And I'll add the concentration below that so I can fact check it later. Okay, let's get started. So now to read the concentration of the standards, we click double-stranded DNA high sensitivity, read standards, we insert standard 1, click read standard. I like to record what I see just in case there's something wrong. And we have 26.91, which is fine. And then we do the same for standard 2. Which is 12059.94. These values are normal for us, but if you have a different device, it might read differently. So now I'm ready to run my samples. I put that I'm adding 4 microliters of DNA. And what I like to do is I like to work in rows of 2, just because of the way the timing is. So I'm going to pipette the working solution into every row, with 2 at a time. And then I'll add 4 microliters of the DNA, vortex it, and then I'll read it because I know about 2 minutes has passed. And then I'll do the same for the next row. Your working time might be a little bit different. Okay, so now I have the concentration of all of my samples, and they look pretty good. Almost all of them are between 6 nanograms per microliter and 12 nanograms per microliter, except for 6. So those 6 samples I will have to redo the pipeline for, and then at the end I'll pool those instead of the ones on this original plate. So that brings us to the end of the pipeline for one plate. However, because my population is over 200 samples and we're only working with 48 samples at a time, I'll have to repeat this at least 4 more times, not including any of the redos. The final stage, which I will not be showing you, is when we normalize and pool our libraries. And what this means is that we have 48 samples on a plate that each have a unique barcode, and all of those samples are going to go into one tube at the end, which will then be run together on one lane on Illumina sequencing platform. So the way that we'll normalize and pool is I would take all of the concentrations that I got from the qubit, use my C1V1 equals C2V2 formula to get one uniform concentration, pipette all that into a new 96 well plate, and then take a set amount from each of those plates and put it into one final Eppendorf tube for each of my plates. And then what I would do is for all of those Eppendorf tubes at the end, which I will have 5 because I have over 200 samples, 48 in each. I will then do another cleanup step just to make sure everything is kosher, where we use the AM Pure XP beads. And then we send it off to the sequencing company and they do a couple of validation tests. And that's the end. Thanks for listening. Hope you learned something about ddRADseq.